Hi friends, good morning. Next video from Dhruva Edutainment and Vidhu classes. Friends, this video from Unit-6, that is Genetics of Intermediate Second Year, friends. What's your question, friends? Describe the steps involved in DNA fingerprinting technology. Very, very important question, friends. Steps involved in DNA fingerprinting technology or protocol of DNA fingerprinting. Friends, DNA fingerprinting. Very, very important question, friends. Friends, DNA. It is deoxyribonucleic acid. It is present in the chromosomes. Okay, friends. This DNA, like our fingerprints, it is unique to person. It is a unique to person, friends. It is unique to a person. Very, very important. Means the DNA of one person is not similar to the DNA of another person, friends. So it is unique to a person. See, no two individuals have identical DNA except identical twins or clones. Friends, identical twins means the twins which are formed from one zygote. They are called as identical twins. And clones, what are clones, friends? The offsprings which are formed by the asexual reproduction are called as what, friends? Clones. So here no two individuals have identical DNA except what, friends? Except identical twins or clones. Okay, friends. Now one more important, friends. All the cells of a person has the same genome. Friends, genome. Genome means what, friends? The haploid set of chromosomes is called as genome. So, all the cells of a person has same genome. There is no change in the DNA of cells of one person. So, all the cells of a person has same genome, DNA sense. All the cells of a person has same DNA. See, friends. DNA fingerprinting is a method used for identifying the individuals by the structure of their DNA. See, friends, in the fingerprint we identify the person with their fingerprint, friends. And what is DNA fingerprinting, friends? In this, we are identifying the individuals by what, friends? By their structure of DNA. By the structure of DNA, here we are identifying the individuals. That is called as what, friends? DNA fingerprinting technology. Like our fingerprints, we are identifying the individuals by their DNA, structure of DNA. That is called as DNA fingerprinting. Okay, friends. This DNA fingerprinting technology discovered by Alec Jeffreys, friends. Very, very important. This DNA fingerprinting technology discovered by whom, friends? Alec Jeffreys, friends. Alec Jeffreys. Once again, Alec Jeffreys. Okay, friends. Now see. It is a technique by which the DNA of an individual can compare with another individual. So why we are using DNA fingerprinting? See, in the DNA fingerprinting, we are comparing the DNA of one individual with another individual, friends. That is, for example, we take suspect in a crime. So we have to compare the DNA of two individuals. One is suspect and the other one is relative or other person, friends, are found in a sample. Okay, friends, that is about the DNA fingerprinting. Friends, what is sample? Friends, what is sample? Please, no need to write this yellowish one, your IPE, friends. Sample means what, friends? DNA sample means a tissue, fluids or other bodily parts of an individual on which DNA analysis can be carried on. So, what is sample, friends? It is a tissue, it is a fluid or a body part of an individual on which we are doing the DNA analysis. Okay, friends. Now, DNA fingerprinting technology has other names, friends. What are they? DNA typing and genetic fingerprinting. Friends, DNA fingerprinting, DNA typing, genetic fingerprinting. These all are same words, synonyms. Friends, DNA fingerprinting, genetic engineering, DNA typing and genetic fingerprinting. These all are same, friends. Now come to steps involved in DNA fingerprinting. First you have to by heart these 8 steps, friends. The first one is obtaining DNA or collection of DNA, friends. Obtaining DNA or collection of DNA. Second one, fragmentation of DNA. Fragmentation of DNA. Third one, separation of DNA. Friends, separation of DNA. Fourth one, denaturation of DNA. Denaturation of DNA. Sense. Fifth one, blotting test. Friends, blotting test. Sixth one, identification of specific DNA by using probes. Identification of specific DNA by using probes. Seventh one, hybridization with probes. Hybridization with probes. Eighth one, friends, exposure on film to make a DNA fingerprinting. So total 8 steps are there, friends. Now we take one by one, friends. Okay, friends, come to first one. What is that, friends? Obtaining DNA or collection of DNA. Afterwards you have to make clones if need or DNA amplification, friends. Now see, friends, obtaining DNA is the first step in a DNA fingerprinting. First you have to collect the DNA, friends. Collect the DNA, obtain the DNA. It is the first step in the DNA fingerprinting. DNA sample is collected from, see friends, blood, saliva, hair roots, friends, not hair. Hair roots, semen, vaginal fluid, buried bones and any tissue, etc. Friends, you may collect DNA from any body tissue, friends, either it is blood or saliva or semen or vaginal fluid or even buried bones also, friends. Okay, that is the collection of the DNA. See, friends, if we needed many copies of DNA, see friends, if we needed many copies of the DNA can be produced by PCR method, friends. So if we won't get sufficient DNA, friends, if we get only 1 or 2 samples, small sample, so you have to make it into number of copies. For that you have to use which method, friends? PCR method. This is for NEET and MCET. By which method we are copying the DNA? What is that, friends? PCR method, friends, PCR method. Friends, here, what is cloning? See, friends, production of many copies of DNA is called cloning or DNA amplification. Friends, here we use one word, friends. What is cloning, friends, here? Cloning means what, friends? Production of many copies of DNA is called as cloning. Cloning is also called as what, friends? DNA amplification. And friends, manaki ఒకటే మనకు డిఎన్ఏ కొంచెమే దొరికిందంటే ఫ్రెండ్స్ మనకు చాలా అవసరం ఉన్నాయి. సో ఈ తక్కువ దొరికిన డిఎన్ఏ నుండి ఎక్కువ డిఎన్ఏ కాపీలను తయారు చేయడాన్ని మనం ఏమంటాం, ఫ్రెండ్స్? క్లోనింగ్ లేదా డిఎన్ఏ యాంప్లిఫికేషన్ అంటాం. సి ఫ్రెండ్స్, కమ్ టు పీసీఆర్. వాట్ ఈజ్ పీసీఆర్, ఫ్రెండ్స్? పాలిమరేజ్ చైన్ రియాక్షన్. ఫ్రెండ్స్, వాట్ ఈజ్ పీసీఆర్? పీ ఫర్ పాలిమరేజ్, సి ఫర్ చైన్, ఆర్ ఫర్ రియాక్షన్. పాలిమరేజ్ చైన్ రియాక్షన్. ఇట్స్ ఏ బయోకెమికల్ టెక్నాలజీ. It amplifies or increases a single or few copies of DNA into many copies. So, PCR is the biochemical reaction in which a single or few copies of DNA is converted into what, friends? Many copies of DNA, some million copies of DNA. This production of many copies of DNA is called cloning or DNA amplification. See, friends, don't write this yellowish one in your IP. Now see, friends, second one, fragmentation of DNA. So now we obtained DNA, friends. Now you have to make it into pieces. You have to make them into fragments. So it is also called restriction digestion. Friends, fragmentation of DNA is also called what, friends? Restriction digestion. So here, why we are calling it restriction digestion? Here we are using some enzymes, friends. They are called as restriction endonucleases. See, friends, in this, DNA is treated with restriction endonuclease enzymes. So this is also called as restriction digestion. Here we are using an enzyme. What is that enzyme, friends? Restriction endonuclease enzyme. Very, very important. So what is its use? This enzyme cut DNA at specific sites, friends. So for fragmentation we are using which enzyme? restriction endonuclease enzyme. What its use? This enzyme cuts DNA at specific sites and forms smaller fragments and forms small pieces. So that means what, when sample is cut into smaller fragments. Now see friends, restriction endonuclease is also called restriction enzyme or DNA cutting enzyme, friends. DNA cutting enzyme. These enzyme recognize and cuts DNA at a particular sites, nucleotide site. Friends, please don't write this in your IP. Now come to third point, friends. Separation of DNA fragments by electrophoresis. Separation of DNA fragments by electrophoresis. See friends, here this is a specific technique, friends, electrophoresis. DNA fragments are separated by electrophoresis. Friends, in the second step, by using restriction endonuclease enzyme, we make number of copies of the DNA. Now we have to separate them according to their size, according to their number of nucleotides. So for this we are using which process, friends? Electrophoresis. So DNA fragments are separated by electrophoresis. In this, used substance is agarose gel. Friends, one more important neat question. Which gel used in electrophoresis? Agarose gel. Friends, agarose gel. Now, electrophoresis is based on their charge and molecular weight. Friends, very, very important. This electrophoresis is based on, friends, charge and molecular weight, friends. Charge and molecular weight. Charge of the DNA, molecular weight of the DNA. Okay, friends. Electrophoresis is based on charge and molecular weight. Now, one more neat question, friends. DNA is negatively charged substance. What is the charge of the DNA, friends? Negatively charged. Very, very important, friends, for MSET and NEET. DNA is negatively charged molecule. Okay, friends. The small pieces of DNA travel farther in the gel. So DNA has small pieces and according to their molecular weight, some are small, some are large, friends. The small pieces move further in the agarose gel, friends. Okay. These DNA fragments form bands in electrophoresis. So these DNA fragments form what, friends? Bands in the electrophoresis. See friends, what is gel electrophoresis? Gel electrophoresis is a technique used to separate DNA fragments according to their size. Okay friends, according to their size. Okay friends, now see friends here, this is gel electrophoresis, friends. See friends, this is a sample, this is positively charged, this is negatively charged. So here DNA shows negative charge, it moves towards the positive charge, friends. So these are the what, friends? Sample, it forms bands. This is what, friends? Gel electrophoresis. Friends, please no need to draw this diagram in your IP test. Okay friends. Now see friends, these are the bands. Some are thick bands, some are the thin bands. See, in the gel electrophoresis we will get bands like this, friends. Okay. See, this is the DNA, friends, negatively charged. It is moving towards what? It is moving towards a positive charge. See friends, here this is a positive electrode. Okay friends. Now come to fourth one, friends. Denaturing of DNA, friends. Denaturing of DNA. Friends, here first you know what is denaturing. You know friends, DNA has two strands. It is double helical in shape. Conversion of two-stranded DNA into single-stranded DNA is called as denaturing, friends. Friends, conversion of two-stranded DNA into a single-stranded DNA is called as denaturing. First, you know the definition of the denaturing, friends. See, friends, denaturing of DNA. See, friends, denaturing means splitting or separation of double helix DNA into a single strand by breaking what, friends? By breaking hydrogen bonds between two strands of DNA. You know, friends, nitrogen bases are bind together by what, friends? Hydrogen bonds. Now, in the DNA naturing, they split the hydrogen bonds. When the hydrogen bonds are splitted by the heat or alkaline chemicals, what happens, friends? The double-stranded DNA becomes a single-stranded DNA. See, friends, here, this is double-stranded DNA. See, they are splitting hydrogen bonds. It becomes what, friends? Two single-stranded DNA. This is called as what, friends? denaturing of DNA. See, DNA on the gel is denatured by using, now friends, DNA is on the electrophoresis gel. On the gel we are using what, friends? Heating or alkaline chemicals. Friends, here heating or alkaline, by heating or by using alkaline chemicals, we are denaturing the DNA. Means what? We are converting the double stranded DNA into two single stranded DNAs. See friends, it makes double stranded DNA into single stranded DNA. See friends, you know the structure of DNA. This is double helix. Here we have splitting, we are splitting the hydrogen bonds. Now it becomes what, friends? Two single stranded DNA. These are what, friends? These are called as what? Complementary strands. You know, remember what one, friends? Complementary strands. Okay, friends. Now come to the fifth one, friends. Blotting technique. Friends, here you know what is blotting, friends? Blotting. Friends, blotting means what, friends? Transfer of DNA molecules from agar gel to a paper or nylon membrane. Friends, blotting means transfer of DNA molecules from agar gel to what, friends? To a paper or to a nylon membrane. Once again, friends, blotting means transfer of DNA molecule from where to where, friends? From agar gel to a paper or a nylon membrane. Okay, friends. Now, it is also called southern blotting technique. Friends, in the DNA fingerprinting, we are using southern blotting technique, friends. We have western blotting, etc., friends. But which technique, which blotting technique is used in DNA fingerprinting? That is southern blotting technique, friends. Once again, NEET question or MCET question. Southern blotting technique. It is discovered by E.M. Southern, friends. It is discovered by E.M. Southern. Through this blotting technique, the DNA fragments on the gel is transferred to nylon membrane by capillary action, friends. One more NEET question, one more MCET. Here, by using capillary action, they are transferring the DNA fragments which are present in the gel onto the what, friends? To the paper or to a nylon membrane. That is called as what, friends? Blotting technique. You are using which blotting technique, friends? Southern blotting technique. Why we are calling it as southern blotting, friends? It is called as, it is discovered by E.M. Southern. Okay, friends. Now come to sixth one, friends. Using probes to identify specific DNA. Friends, what is probe? Probe is a single stranded DNA. It is known, a probe is a known single stranded DNA. We know the nitrogen basis of that DNA. So what is probe? Very, very important. It is a single stranded DNA. Which the single strand DNA, friends? Known single strand DNA. Known means what, friends? We know the arrangement of the nitrogen base of that DNA. Okay, friends. So what is probe? A probe is a known single stranded DNA, friends. It is only single strand. It has no double stranded. Probe is a known single stranded DNA. This probe is used to search for its complementary sequence in a sample. You know, friends, we have nitrogen bases. These nitrogen bases are having the form the base pairs. So the nitrogen base which are present in the probe form a pair with the nitrogen base, complementary sequence of the DNA, friends. So this probe is used to search for its complementary sequence in a sample. Okay, these are radioactive probes, friends. These probes are what, friends? Radioactive probes. Means what, friends? They are labeled with what? Radioactive elements. These probes are radioactive or fluorescent probes, friends. Radioactive or fluorescent probes. Now come to hybridization with probe. See, friends, in this hybridization means quite opposite to denaturing. In denaturing, double stranded DNA becomes single stranded, friends. In hybridization, single stranded DNA becomes double stranded. Okay, friends. Now see, friends, hybridization means it is a process in which two complementary single stranded DNA molecules bond together form double stranded molecule. See, friends, in DNA, in denaturing, what happens? double stranded DNA convert into single stranded. In hybridization, two single stranded DNAs convert into what friends? One double stranded DNA. So what is hybridization? It is a process in which two complementary single stranded DNA molecules bond together by hydrogen bonds and form a double stranded molecule. That is called as what friends? Hybridization. Please don't write this in your IP friends. See friends, in DNA fingerprinting technology, single stranded radioactive DNA probe is used for hybridization of DNA fragment. Friends, here we are using what friends? Radioactive DNA probes or fluorescent DNA probes. For what? For hybridization. Okay friends. After the probe hybridizes, so the hybridization takes place friends. Now we have excess DNA. The excess probe means the probe which are not matched is washed up by washing. So we used the probe friends. Some of the probes matched with the sample DNA. So there is remaining probe which is not matched or unmatched. That excess probe is washed by what friends? By washing. Okay friends. Now, a photographic film is placed on the membrane containing DNA hybrids. Now, next step friends. A photographic film is placed on the membrane containing the DNA hybrids. See friends, this is what is friends? Hybridization. Radioactive probe, single stranded probe, this is complementary probe friends. Now it is forming what friends? This is double stranded DNA. This is hybridization. Quite opposite to denaturing. Okay friends. Now last one friends. Exposure on film to make a DNA fingerprint. When the radioactive probe is exposed towards X-ray film, it forms an image of bands that are identical to specific DNAs. Friends, so when the radioactive probe is exposed towards X-ray film, it forms an image of bands, friends. It forms an image of bands that are identical to specific DNA bands. Okay, friends. Thick and thin bands are obtained, which forms a pattern of bars which constitute DNA fingerprint. See, friends, thick and thin bands are formed. See, friends, this is here we have one example, friends. See, friends, mom, dad, D1, D2, S1, S2. D1 means daughter 1, friends. D2 means daughter 2. S1 means son 1, son 2, friends. See, friends, here these are wife and husband, friends. See, D1, daughter 1 is their own daughter. D2 is the stepdaughter. S1, own daughter. S2 is not related, friends. He is the adopted son. Now you see, friends, see this S2 has no bonds with these two parents. See, here all green bands are there. See, friends, here we don't have those green bands. Now see, this D1 is their own daughter. See, here it has similar bands. D2, friends, D2 has the bands of the mother only. She has no bands of the father. Means this daughter is a stepdaughter. Okay, friends. Now here S1. S1 is their own son. See, he has the same bands. Okay, friends. This is about what, friends? DNA fingerprinting. Okay, friends. DNA fingerprinting technology. Thank you, friends. Good day. Very, very important question, friends.